So we're just going to start on time, and we're very pleased to have Keiichiro Mine here. He's at the biology department at Boston College in the lab of Emma Altinus, and his talk today is the role of tyrosine kinase 2 or TYK2 in the pathogenesis of T1D, type 1 diabetes. And a little bit about him: he received his bachelor's and master's degree in medical technology from Kyushu University in Japan. He studied the pathogenesis of virus-induced diabetes and the role of gamma delta T cells in inflammation at the Medical Institute of Bioregulation, Kyushu University, Japan, where he received his PhD in immunology in 2020. And after completing his PhD, he worked as a postdoctoral fellow and a project assistant professor at Sagi University, Japan. His studies revealed the diverse role of TYK2 in the pathogenesis of viral-induced and autoimmune diabetes, and he joined the Altinus lab in April 2024 to study the role of gut microbiota in autoimmune type 1 diabetes. His recent paper is a Nature Communications paper. It's a great paper. And I'm going to drop it in the chat right now. And thank you so much for joining us, Keiichiro. I'm really excited to hear this talk.
Thank you for the kind introduction, Monica. It's my great pleasure to be here today, and I'd like to thank the organizing to talk about our research related to tyrosine kinase 2, TYK2. My research focuses on how type 1 diabetes is caused by environmental factors such as viral infections and also by autoimmune mechanisms. First of all, I'd like to talk about our target molecule. Tyrosine kinase 2, TYK2, belong to the Janus kinase family and plays a role in cytokine signaling, including type 1 interferons, IL-10 and IL-12. When these cytokines bind to the cell surface receptors, TYK2 is activated, phosphorylates the intracellular tail of the cytokine receptors, and recruits STATs in concert with other JAKs. These recruited STATs are phosphorylated and translocated to the nucleus to regulate cytokine-specific gene expression and cytokine secretion. TYK2 is important for antiviral and antibacterial defense. However, TYK2 is also involved in the pathogenesis of autoimmune diseases and is a potential therapeutic target for them. Initial genome-wide studies reported that the locus where the TYK2 gene is located is associated with type 1 diabetes. Recent studies, more narrowed down, reported that the loss of function SNPs in TYK2 genes has protective effect in type 1 diabetes and also as autoimmune diseases. In contrast to these studies, we reported that SNPs at TYK2 gene promoter region are a risk factor for type 1 diabetes, particularly in patients associated with flu-like syndrome at the onset in the Japanese population. Some papers have reported the role of TYK2 in beta cells. In this paper, in human beta cells, TYK2 inhibition prevented the mimic of viral RNA-induced apoptosis. The other paper showed the loss of TYK2 is associated with the defective formation of endocrine precursors from iPS cells. These results suggest that TYK2 signaling is associated with inflammation and cell death in beta cells, and also potentially associated with the development of beta cells. We have studied the role of TYK2 in type 1 diabetes using animal models and published two papers. One is virus-induced diabetes, and the other is autoimmune diabetes. I'll first introduce the study using virus-induced diabetes model and then talk about the study using the autoimmune model. As you know, viruses may trigger beta cell destruction in several ways: direct destruction, induction of beta cell death or dedifferentiation in several ways, and triggering alert autoimmunity. There are some available animal models for virus-induced or virus-triggered diabetes. Encephalomyocarditis virus, this strain is highly beta cell tropic and replicate in beta cells and induce diabetes in mice within 5 days after infection. This is a model of direct destruction of beta cells, but also macrophage also plays a role in this model. Kilham rat virus and rat insulin promoter derived LCM virus glycoprotein transgenic mice model developed diabetes within a few weeks after viral challenge by the autoimmune mechanisms. We analyze the mechanisms of direct viral destruction using EMCD virus. EMCD virus belongs to the picolan-virulate family and is a non-enveloped RNA virus. In 1968, it was first reported that the EMC virus could induce diabetes in inbred mice. And subsequently, a highly diabetogenic EMCD virus was isolated. EMCD virus could induce diabetes in some strains of inbred mice, such as DBH mice, within 5 days after infection, but not in Black 6 mice. This virus rapidly replicates in the pancreas of susceptible strains of mice within 3 days after infection, suggesting that early time point after infection is important for the development of EMCD virus-induced diabetes. The innate immune system recognizes viral infection using pattern recognition receptors and produce type I interferons. Type I interferons enhance the antiviral state through JAK-STAT signaling. It was already reported that the toll-like receptor 3 and IRF3 and interferon beta-1 deficient mice developed diabetes after EMCD virus infection. We focus on TYK2 because, as I mentioned before, some studies suggested that TYK2 gene locus was associated with type 1 diabetes in humans. We generated TYK2 gene knockout Black 6 mice and revealed that the mice developed diabetes after EMCD virus infection. So we analyzed the role of TYK2 in virus-induced diabetes using this model. This is a highlight of this study. We generated transgenic mice carrying mouse insulin promoter with mouse TYK2 gene and crossed with TYK2 knockout mice. This beta cell specific TYK2 gene expression mice didn't develop diabetes after EMCD virus infection. So this result suggests that TYK2 gene in TYK2 expression in beta cells is critical to prevent EMCD virus-induced diabetes. To reveal why TYK2 knockout mice develop diabetes, we performed ex vivo cell viability assays. Mouse embryonic fibroblast and islet cells were analyzed. We found that the high concentrations of interferon beta prevented cell death by EMCD virus infection in Treg TYK2 knockout mouse embryonic fibroblasts, but not in the islet TYK2 knockout islet cells. In addition, mouse insulin promoter derived TYK2 expression in beta cells restores the survival of islet cells after EMCD virus infection in the high concentrations of interferon beta. These results suggest that impaired responses to type I interferons and antiviral difference in beta cells due to TYK2 knockout may be associated with diabetes onset after EMCD virus infection. Based on these results, we concluded that the reduced TYK2 expression is a risk factor for beta cell tropic virus-induced diabetes. However, the precise role of TYK2 in autoimmune type 1 diabetes has not been reported. So I studied to analyze the role of TYK2 in autoimmune diabetes using NOD mice. My question was, is the role of TYK2 in type 1 diabetes dependent on causal factors? To this end, we generated TYK2 knockout NOD mice and found that the loss of TYK2 suppressed the development of autoimmune diabetes in NOD mice. Diabetes incidence was comparable between wild type and TYK2 hetero knockout mice. Insulitis was suppressed in TYK2 knockout NOD mice. Next, to determine whether TYK2 expression is important in immune cells or the environment, we performed transfer experiments. NOD-skipped mice transferred with TYK2 hetero knockout splenocytes developed diabetes, but mice receiving TYK2 knockout splenocyte had reduced diabetes incidence. These results suggest that TYK2 expression in immune cells is important for the development of autoimmune type 1 diabetes. Next, we generated TYK2 knockout NOD3 mice and revealed that the loss of TYK2 in the host environment led to reduced diabetes incidence induced by transferred splenocytes. These results suggest that TYK2 expression in immune cells and in the host environment both contribute to the development of autoimmune type 1 diabetes. So we analyze immune cells and beta cells and reveal that TYK2 signaling plays a role in the three types of cells: pancreatic beta cells, CD8 positive resilient type dendritic cells, and CD8 positive T cells. First, we focus on beta cells because we thought beta cells are important components of the host environment. Beta cells are purified and analyze gene expression profile. PCA reveals that beta cells from 6-week-old TYK2 wild-type mice and TYK2 knockout mice had similar gene expression profiles, but beta cells from 11-week mice had different gene expression profile from those of 6-week mice, suggesting that TYK2 genotype affects the gene expression profile with aging. Normalized and selected gene expression levels are shown in the right side. Chemokines such as CXCL10 and CXCL9 and interferon-stimulated genes were slightly lower in beta cells from 11 weeks old TYK2 knockout compared to beta cells from 11-week TYK2 wild-type beta cells. Importantly, the expression levels of FAS death ligand was lower in beta cells in 11-week TYK2 knockout compared to 11-week TYK2 wild-type beta cells. These results suggest that inflammatory responses in beta cells with age are attenuated in TYK2 knockout mice. We next focused on the immune cells. To analyze immune cells in the pancreas, collagenase solution was injected into the pancreas through the bile duct. I think maybe because pancreas is rich in digestion enzymes, so some collagenase types were not good for the preservation of epitope integrity on immune cells. We found that the combination of collagenase D and trypsin inhibitor was good for analyzing immune cells in the pancreas. And we use this method to analyze immune cells in the pancreas. The number of CD8 T cells in the pancreas of TYK2 knockout mice was decreased compared to TYK2 sufficient mice. However, the number of CD8 T cells in the pancreatic lymph node was comparable between genotypes. Other types of immune cells also showed the same kinetics. We analyzed the phenotype of T cells and found that the naive CD8 T cells were increased in the pancreatic lymph node of TYK2 knockout NOD mice, but not in the pancreas. This was not observed in CD4 T cells. We further analyzed phenotype of CD8 T cells in the pancreatic lymph node and found that interferon gamma producing CD8 T cells were decreased in the pancreatic lymph node of TYK2 knockout NOD mice. This reduction was not observed in CD4 T cells. These results suggest that the defective proliferation and or development of CD8 positive cytotoxic T lymphocyte in the pancreatic lymph node are associated with reduced diabetes incidence in TYK2 knockout NOD mice. So next we focused on CD8 T cells and performed transfer experiments using islet-specific antigen, IGRP-specific CD8 T cells. Naive CD8 T cells are primed by dendritic cells through MHC class 1. This process is called cross-priming. The transferred naive TYK2 heteroknockout or TYK2 knockout CD8 T cells into TYK2 heteroknockout recipient mice resulted in proliferation in the pancreatic lymph node 5 days after transfer. However, their proliferation was suppressed in the pancreatic lymph node of age-matched TYK2 knockout recipient mice. These results suggest that CD8 T cells' extrinsic mechanisms have a role in the proliferation of islet autoreactive cytotoxic T cells in the pancreatic lymph node. So we next analyze dendritic cells, which have a central role in the adaptive immune system and cross-priming. We found that TYK2 deficiency reduced the expression of MHC class 1, a crucial molecule for presenting antigen to the CD8 T cells, and CD8 positive resident type dendritic cells, but not as a subset of dendritic cells. TYK2 knockout CD8 positive resident DC showed reduced cross-priming of CD8 T cells compared to TYK2 wild type CD8 positive resident DC. These results suggest that TYK2 knockout CD8 positive resident DC has impaired APC function, maybe due to reduced expression of MHC class 1. To summarize the first part, in the TYK2 knockout mice, inflammation in beta cells is reduced, and in the pancreatic lymph node, CD8 positive resident DC has impaired cross-priming capacity, and the number of naive CD8 T cells is increased. Our data reveals that the cross-priming of CD8 T cells is impaired, but not abolished. This means cytotoxic T lymphocytes are generated even in the TYK2 knockout mice. So next we characterize the antigen-experienced CD8 T cells that have CD44 high phenotype. Because the transcription factors are key regulators of T cell effector functions, we examined some transcription factors and found that CD44 high CD8 T cells positive for TBET were decreased in the pancreatic lymph node of TYK2 knockout mice. The expression levels of TBET were also reduced in CD8 positive T cells. IL-12 is well known to induce TBET expression in CD4 positive T cells. So we therefore focused on the IL-12 TYK2 signaling axis. When the CD8 T cells were stimulated with IL-12, the upregulation of TBET expression levels was suppressed in TYK2 knockout CD8 T cells, suggesting that TYK2 knockout CD8 T cells develop cytotoxic T lymphocyte with lower expression levels of TBET due to defective CD8 T cells intrinsic IL-12 signaling. TBET is an important transcription factor for the production of interferon gamma and the expression of chemical receptors, CXCL3, a receptor for CXCL9 and CXCL10. CXCL3 is important for the T cell migration to the inflammatory site. We found that CXCL3 positive CD8 T cells were decreased in the pancreatic lymph node of TYK2 knockout mice, suggesting that the migration of CD8 T cells to the pancreas was reduced. These results suggest that the decrease in the number of CD8 T cells in the pancreas of TYK2 knockout mice may be due to reduced in the number of TBET positive and CXCL3 expressing CD8 T cells in the pancreatic lymph node. Next, to reveal the effective function in CD8 T cells, we performed RNA sequencing analysis on purified antigen-experienced IGRP-specific CD8 T cells in the pancreatic lymph node and pancreas. The DNA expression profiles of the cells in the pancreas differed from those in the pancreatic lymph node, regardless of the TYK2 gene genotype. However, we found that the effector molecules, granzyme B and facel, were upregulated in TYK2 wild type CD8 T cells in the pancreas compared to those of TYK2 knockout cells. This result suggests that the loss of TYK2 impairs the upregulation of effector molecules in islet autoreactive CD8 T cells in the pancreas. To reveal the mechanisms of impaired upregulation of effector molecules in CD8 T cells, we performed ex vivo co-culture experiments. After priming the IGRP-specific CD8 T cell, the expression levels of granzyme B in the cells were comparable between genotypes. However, the TYK2 sufficient CD8 T cells cultures with NIT1 cells, pancreatic beta cell lines, had upregulated expression levels of granzyme B. This was attenuated by TYK2 deficiency and by the blockade of interferon beta signaling in CD8 T cells. The expression levels of interferon beta 1 gene in NIT1 cells were rapidly upregulated after co-culturing with CD8 T cells, suggesting that the source of interferon beta is pancreatic beta cells. These results suggest that interferon beta signaling was associated with upregulation of granzyme B in CD8 T cells after encountering target cells. Indeed, the killing capacity against NIT1 cells was reduced in TYK2 knockout CD8 T cells compared to TYK2 hetero knockout CD8 T cells. Together, these results suggest that the upregulation of effector molecules after encountering target cells is impaired in TYK2 knockout CD8 T cells. Finally, to test the effect of TYK2 inhibitor, purified CD8 T cells were stimulated with ex vivo in the presence or absence of TYK2 inhibitor. The expression levels of TBET and stimulated CD8 T cells were reduced, and the beta cell killing capacity of the CD8 T cells were impaired TYK2 inhibitor dose dependently. Expression of chemokines and interferon stimulated gene in beta cells induced by type 1 interferons was suppressed by TYK2 inhibitor dose dependently. NOD wild-type mice treated with TYK2 inhibitor by oral garbage once daily for 4 weeks from 6 weeks old of age had reduced diabetes incidence compared to vigor treated mice. In contrast, treatment with TYK2 inhibitor at the same dose from 12 weeks of age resulted in comparable diabetes incidence with big control mice. The low sample size is our limitation, but these results suggest that the treatment with TYK2 inhibitor studied at an early stage of type 1 diabetes may be a potent strategy to prevent the development of autoimmune type 1 diabetes. In summary, our studies reveal that TYK2 has a Janus phase role in type 1 diabetes. In the autoimmune type 1 diabetes, loss of TYK2 prevents the development of type 1 diabetes by suppressing acquired immunity against beta cells and inflammation in beta cells. In contrast, in beta cell tropic virus-induced diabetes, loss of TYK2 is a risk factor for the development of type 1 diabetes because of reduced antiviral difference in beta cells. A recent preprint reported the effect of TYK2 inhibitor in NOD mice with enough sample size, good study. In agreement with our findings, the authors also show the protective effect of TYK2 inhibitor in autoimmune type 1 diabetes. In addition, the authors employed a virus-induced autoimmune diabetes model, RIP LCM virus GP mice model. These mice expressed LCM virus glycoprotein in beta cells, and after LCM virus challenge, developed LCM virus specific T cells attack beta cells. The authors show that TYK2 inhibitor treatment prevented diabetes development in this virus-induced autoimmune diabetes model. In summary, our studies reveal that the role of TYK2 in type 1 diabetes is dependent on causal factors. Reduced TYK2 gene expression is a risk factor for beta cell tropic virus-induced diabetes. However, in the context of autoimmune diabetes, TYK2 signaling promotes the development of autoreactive CD8 positive cytotoxicity lymphocyte and inflammation in beta cells. In addition, a recent paper adds insight into virus-induced autoimmune diabetes. From our studies, we believe that the advantages and risk of immunomodulation should be considered when developing treatment for type 1 diabetes. I'd like to thank all collaborators and fans. I'm happy to take questions. Thank you very much for your attention.
Thank you very much, Keiichiro. Just to note, Farouk Syed, one of the author, or the primary author of the paper you referenced, is on the call, so maybe he'd like to chat with you a little bit. And I also just wanted to give a shout out here to the fact that deucravacitinib, the TYK2 inhibitor that was used in both of those papers, was a BMS drug that was released in 2022 for moderate to severe plaque psoriasis. It was the first innovation in oral treatment in 10 years. And it was fantastic that you took that product and, set about testing in the context of type 1 diabetes. Here's a couple of questions in the chat. Richard Coffey, to everyone, have you ever observed diabetes development in the MIP TYK2 transgenic mice without viral infection?
Thank you for the question. And we didn't observe diabetes onset in the MIP TYK2 transgenic mice.
Okay. And then from Kiadi Gerder, have you studied which organ the TYK2 inhibitor was acting on in mice? You showed effects on both beta cells and immune cells after TYK2 inhibition. Can you clarify where the inhibitor is having its primary effect?
We treated TYK2 inhibitor by oral garbage, so we believe the TYK2 inhibition was occurred in all cell types in whole bodies. So after the treatment of TYK2 inhibitor, the autoimmune responses were suppressed in the immune cells and also beta cells. And also the other organs such as lymph node should be occurred, and all of them were orchestrated to suppress the development of type 1 diabetes.
Yeah, this is really interesting. It seems as if, your group and the Indiana group have sort of just, waded into this new approach. And it's really going to be interesting as these, as further studies develop. I just had one question for you too before we go. And that is, how are you going to now, I took some notes here, how are you going to be tying in your microbiome approach in the Altinus lab with this previous research that you have presented today? Or is it totally separate?
Yeah, some relation. TYK2 is associated with IL-23 signaling. It's very important cytokines in the gut immunity. So the microbiome signaling, I think, is associated with TYK2 signaling. So I hope I could connect with the gut microbiome signaling to the TYK2 in the future, but I'm not sure if this molecule is truly associated with the gut microbiome and type 1 diabetes onset.
Yeah, well, there's a lot of microbiome education of CD8 positive and CD8 positive resident DCs in the PLN. So there's, there's some food for thought there to kind of investigate that. Okay, last call for questions.
Hi, Monica, this is Farouk.
Hi, Farouk, how are you?
I'm good. How are you?
Great.
That's great. Mine, I have a question. You guys have knocked out the entire locus of TYK2 or just add the mutation of the humans, the 356 mutation that showed to have protective effect in Japanese population? So have you guys tried just having that 356 mutation in your mouse model where you inserted the human SNP to see what's the real effect comparing to when you fully knocked out that SNP?
Thank you for the question. And yeah, we should do such experiments, knocking experiments, but we never did such a experiment in vivo.
Have you guys ever tried to look into the exhaustion markers? Because JAK inhibitors showed some exhaustion, teplizumab showed exhaustion of T cells, right? Even in our study with TYK2, we have seen CD8 cells exhaustion as well as activation of FOXP3 positive cells with PD-1 positive. So I was just wondering, have you guys also looked into the other side, like how this might be working with the immune system as Giridhar asked? It's similar to, like, we have seen both the sides, both beta cell specific differences as well as immune cell specific difference in different organs.
We check the PD-1 expression in the immune cells, and there are no differences between the TYK2 genotype. And also we analyze the FOXP3 positive CD4 T cells, Tregs, and their number of FOXP3 positive CD4 T cells were comparable between genotypes. So
That's interesting.
yes.
Thank you.
Sounds like a conversation to be continued between the two of you. There's a great resource there at Indiana, and of course the Altinus lab is so highly regarded as well. So hope that will continue offline. In the meantime, thanks everyone for joining. This was a really interesting talk. And we also had Farouk Syed earlier this month. So those two talks were well worth listening to. And I hope everyone has a great rest of your day. Thanks again, Keiichiro.
Thank you very much.
Have a great rest of your day. Bye-bye.
Bye.